Hello, everyone. Welcome to the 2023 virtual meeting of the Phytophthoras and Native Habitats Work Group, also known as CalPhytos. We're very happy to have you all here together to listen to this year's developments. As a reminder, I just mentioned we are recording this meeting for those who cannot be joining us live. I'm Janice Alexander from the University of California Cooperative Extension in Marin County, one of your hosts for today. Joining me as moderators are two co-leaders of our Phytophthoras work group, Diana Benner of the Watershed Nursery and Mia Angolia of the San Francisco Public Utilities Commission. Helping with tech in the background is Bonnie Nielsen, also from UCCE Marin. We have a number of great speakers and topics this morning. As each speaker gives their presentation, you're invited to submit comments and questions in the chat. I'll be assisting getting those questions over to the moderators and the speakers so that they're able to address them at the end of each talk. That's the background information you guys need logistically for the meeting. And so I'm going to get out of the way and hand this back over to Diana Benner to get us started with the official talks. Thanks, Diana.
All right. Thank you. Good morning, everybody. And thank you, Janice. And thanks, everybody who's here for your interest. I am delighted to be able to introduce Dr. Schiffer Forsyth, who's going to be presenting this morning on nursery green waste and potential risk factors for nursery plants, as well as the larger environment. So take it away.
Hello, thank you. I hope everyone can hear me. Okay, can you see my slides? Yes, okay. So not quite doctor yet, I'm still a student, but thank you for introducing me. I'm Kadiatou Schiffer Forsyth, and I'm very happy to be presenting to you work I did while I was at the University of Edinburgh with forest research with Sarah Green, who should be here in the meeting as well, and Debbie Fredrickson-Matika. And the project was on looking at the risk from Phytophthora in green waste produced by nurseries in Scotland. So I probably don't need to explain this to the audience we have here today, but nursery stock can harbour Phytophthora species that can be highly diverse. And Phytophthora as a soil-borne pathogen is very prone to spread through the trade and in nursery systems. And a well-established route of spread into native habitats or forestry is through the outplanting of nursery stock. So obviously we need to limit that. And so there are well-established routes of spread through outplanting, for example, and you'll probably be able to tell me about many more, but through footwear, movement of people, water runoff, contaminated machinery, etc. But a route that's maybe not mentioned often is direct spread from waste into the wider environment, which has been observed to be a potential problem in nurseries across the UK. And so these kind of piles are found in many nurseries throughout the country and could be a place where Phytophthora aggregate because these piles are made up of culled stock plants, hedge cuttings, all kinds of waste materials that kind of just hang around for a while and are sometimes not stored appropriately to limit the risk of spread of pathogens. So this project set out to determine what Phytophthora species are present in some of these waste piles and whether they could pose a risk to the wider environment. And this would then provide diagnostic evidence for guidance on the safe handling of horticultural nursery green waste if there is a risk of it containing pathogens. So yeah, essentially, are there Phytophthora in the waste piles, and are these a risk of spreading back into nursery systems, into the wider environment, or the forestry sector? I could also include horticultural production systems here, I guess. And so here's an overview of our workflow. So we sampled 3 nurseries in central Scotland, and we took water samples, solid waste samples and root samples. I'll explain what that means in a moment. And we analyzed these using 2 different methods, baiting and metabarcoding, both resulting in species identification. So we took the solid waste samples from 3 different levels just to cover the kind of depth of the piles, so at different depths, and Then the water samples were taken from puddles if they were present. If not, we used the flow-through method, pouring water on coarse material and then filtering the water to using these modified knapsack sprayers, maybe a system you're familiar with, probably actually. And then we also took root samples directly from culled stock plants where they were present. And so the metabarcoding method involves extracting environmental DNA from the samples and then amplifying the target region specific to Phytophthora. Here it was the ITS1 barcode region, and then sequencing that barcode region that would then allow the identification of the species. And so this whole bioinformatics part was done at the James Hutton Institute in Dundee, and species identification was assigned using a database that had been previously developed in the THABI-PICT project by Peter Cock, David Cook, Sarah Green, and so between Dundee and Forest Research. For the baiting, that was only possible for the solid waste and water samples, not for the root samples directly, but you'll probably be familiar with the principle. But yeah, zoospores are attracted to bait leaves that are floated on the water surface. And then the little grey lesions that appear, as you can see on this last image, are then transferred onto selected media, and the Phytophthora cultures that grow from it were then sequenced to allow their identification. So let's come to the results. We found 18 Phytophthora species. That's combining all three nurseries we sampled, all three sample types and the two methods. And here they're listed in alphabetical order. I've highlighted in red the ones that were the 8 species that were obtained baiting, so of which live isolates were obtained. The rest were only detected in metabarcoding. And so these 18 samples were, 18 species, sorry, were found in only 2 of the nurseries. One of the nurseries, there was no Phytophthora at all, as far as we could tell in the waste. And so I must say as well that the 3 nurseries we sampled were selected because they would represent a broad spectrum of different management practices. So kind of from a specialist horticulture nursery all the way to commercial nurseries that import most of their stock. So this first nursery where we didn't find any Phytophthora was a non-commercial specialist nursery where 99% of stock is propagated on site. Very little is imported, and if it is, then there's a quarantine area, and they have robust biosecurity measures in place that they observe. And the waste is treated in a very rigorous process with composting. It's held on in a concrete holding area, and it's composted in a way to ensure that pathogens are killed, so by monitoring temperature and moisture content. Yeah, so the second nursery is where we found 18 Phytophthora species, so all of the ones I've listed previously. And this was a commercial nursery where all of the stock was imported essentially from the UK and the EU. But I guess, yes, sometimes tracing that back exactly is quite difficult. And in this situation, waste was stored at the back of the nursery site near a stream and hedgerow. And the third nursery we sampled, we could detect 9 Phytophthora species. It was also a commercial nursery, although some stock was also propagated on site. So this is just to show that the 3 sample types, we took the solid waste, roots and water samples, enabled us to kind of get a more complete picture of what Phytophthora would be present, and also shows that many species are present in water, which is runoff and which visibly leaches from the piles into puddles and potentially into streams and into the wider environment. So that was quite a relevant finding, I think. And so this Venn diagram is just comparing what species were detected in metabarcoding and in baiting. So it was expected that there wouldn't be a complete overlap, of course, because metabarcoding is very, very sensitive. And in baiting, not all species can be recovered due to different growth rates and preferences that can't be replicated in the lab, I guess. But so the advantage of metabarcoding is that it gives a more complete picture, but baiting also provides direct evidence that these species are present live and can grow from the material we sampled. And so these are pictures of the cultures that we obtained. So these are isolates from the 8 species that we baited, and including Phytophthora ramorum, which is a quarantine pest in the UK and is obviously a big concern. So in parallel to this project, other work funded by the Plant Health Centre aimed to kind of understand what the situation is with most nurseries in the UK and how waste is treated and managed at the moment. So as part of one of the workshops, this question was asked, what do you do with waste plants and plant material? And these workshops really aim to understand what is going on and what measures could be put in place to incentivize and help nurseries to manage their waste differently if it was a problem. And so here are the responses. So yeah, 41% of nurseries said that they have a waste pile on site. 18% send their material to landfill, and 6 responded other, so that can mean a range of things. But the point is that most nurseries have this situation of having a waste pile. And 83% of respondents felt that their current waste management practices needed improvement. So there is definitely the will to do something about this. And so through this project, we could show that in nurseries where Phytophthora is present in stock plants, it's kind of common sense, but it obviously ends up in the waste, and it will stay in the waste unless it is heat treated in a way or composted efficiently. And so this guidance document was produced by the Plant Health Centre and the Scottish Government, and so that's available to nursery managers, and it kind of sets out best practice related to the waste management. And here are 2 photos of kind of effective composting system at the top, and then the second kind of one of the important points in the guidance was to have concrete holding areas to prevent runoff and have separate bays to treat material appropriately and differently according to what it is, and especially treating high-risk material separately from the rest. But the guidance especially mostly focuses on avoiding Phytophthora in the nursery setting in the first place, because obviously you have to better to tackle the problem at the source further upstream rather than at the end when it's already in the waste. So if you want to know more about this work, we published it early this year in this article, Phytophthora in Horticultural Nursery Green Waste. And you can access it for free just through this QR code, and the Plant Health Centre guidance is available through the other QR code, the second one. And so thank you for your interest, and I look forward to your questions and comments.
Okay, that was fascinating. Thank you so much. We have, as Janice said, you are welcome to put questions. They're in the chat. Great work, and yeah, management all the way back from the potato famine epidemic time. Please feel free to add questions there. In the meantime, I have some that I'd like to ask you in regards to, I mean, my thought initially when you were talking is wonderful that you have this document available to nurseries to give support of, okay, so what do we do? Because everyone's going to have green waste. So providing guidance on the composting effectively and efficiently on site is wonderful. And yeah, so someone had a question about, or a comment about P. ramorum. And I was a little surprised. I'm a nursery person. I don't know anything about this, but surprised that the meta detection method didn't capture the P. ramorum. Is that expected?
Yes, well, it was a bit of a surprise to us well, first of all, to find ramorum at all, and then that it didn't come up in the meta barcoding. But that could be due to one of the limitations of meta barcoding is that the material you use to start in the process is very, very small. So you extract the DNA from only a few milligrams of soil. And whereas with the baiting, we had about 250 to 500 grams of material each time. So it could just be that propagules didn't happen not to be in the sample we used for the metabarcoding, for example. So that's one of the possible explanations for that.
And then we have a question of, can you describe how the water was flushed out of the waste before baiting?
Yes, okay, sorry. So for the baiting, we took water directly from puddles. We just kind of scooped it up with a modified milk container, milk bottle. And then we just put it in bottles, brought it to the lab and put that in the containers as it was. And for the solid waste samples for baiting, we just put those in the bottom of the tub and then flooded it with sterile water because of the zoospores are obviously motile in water, and so they would swim up through the water to infect the bait leaves. I hope that answers the question.
I have a question about why leaves were used instead of pears.
I guess that's just what the lab was using and the experience we had, that worked quite well. I have tried pears, but I've not had success with it really. Yeah, and so what species of plants were used for the bait leaves kind of tried to cover a mix, but it was limited by the period of time during which the project was done. It was kind of the end of winter, so we had to take evergreen species. So we had Quercus ilex, rhododendron, hebe, and ivy as well, heterohelix.
Another thing that came up in my mind when you're talking about this, in the document you're producing, you've produced the containment of any runoff from the compost piles, but it also brought to my mind thinking of, before that material becomes green waste, moved there, it's potentially a vector sitting in the production area. So are you going to do any looking at sort of making hedgerows or catchment systems around nursery production areas?
I don't. Maybe that would be a way of approaching it, but I guess the separating waste according to risk level. So if you have culled stock plants that are culled because of suspected Phytophthora infection, for example, definitely to handle those separately from any other waste that might not be contaminated, just kind of reduce the volumes that you have to then deal with. But yeah, it was in previous work that was done by people at Forest Research with the Phytophthoras project. They did see kind of hedgerows next to nurseries, trees in them showing symptoms of Phytophthora infection. So it is definitely important to keep that waste away from the natural environment as well as the nursery stock. Yeah.
Okay, I have more questions, but somebody else put in here, can we assume that these waste piles did not compost on their own? And how is this checked?
Okay, that's a good question. It wasn't really checked or measured in any way in this project, but just I think the kind of time that these piles are around and the nature of the material there makes us think that it probably wouldn't compost. Lots of big twigs and just not enough organic material to get the process going really. And in situations like in the first nursery where there was an effective composting system, you could really tell just from the heat that was produced by the pile, you could feel it with your hands that there was effective composting in place. And also the workers at the nursery said that, I mean, they turn it regularly. That was quite obvious as well. So there is a monitoring process of the composting.
All right, that's great. There's definitely more to discuss on that, like buying material in. But thank you so much.
Thank you very much.
Maybe we'll have more time for questions later. But at this moment, I would like to welcome Dr. Sweeky, Dr. Beret, and Dr. Frankel to present on the highly anticipated new diagnostic guide.
All right. Thanks. All right. Let me make sure we're sharing properly. You're probably seeing the main screen now. Is that right? Let's see.
Yep, Ted, we can see the main screen.
Okay. Just make it Get this bar out of the way. And which view do you have there?
We can see the regular slideshow.
Okay, so you're all set in the regular slideshow. Great. Okay. We're going to talk a little bit about this new diagnostic guide for sampling for Phytophthora restoration areas. The authors are shown here, myself, Elizabeth Bernhardt, Tyler, and Susan Frankel. But Susan and Tyler aren't to blame for this presentation. I pretty much put that together myself. So we'll go on. I'm not going to talk a ton about the publication. We have a lot of new faces out in the group out there. And so one thing I wanted to start with is to actually set some context for this, as well as provide some background so we kind of and really see the utilization and how this is intended to be used. So I'm going to take this back in time to 2014, which we can refer to perhaps as the Great Soil Phytophthora Awakening. And this was a year when there were several things that happened. One of those was some sampling occurred at some very large restoration projects. And one of the first samples pulled there and sent into us was this toyon here, which had really completely rotted roots. It died shortly after it was planted. Happened to have both Phytophthora cactorum and tentaculata on it. And tentaculata is a species that has some regulatory significance because it's not really been found in the US more than once before and was not considered to be established. So that got people kind of excited about soil phytophthora in a way they might not otherwise have been. That initiated quite a bit of sampling at a number of different locations associated with this very large restoration project, which entailed, as you can see from this, the dots there, sampling locations, digging up huge numbers of samples and testing those for Phytophthora presence. Now, as it turned out, also in 2014, we got a call on another project that was assisted migration planting of endangered species. There were some issues with the plants from the start of that project, and we found that by sampling them, that they were also infested with Phytophthora cactorum, and almost universally, it was almost every plant in the entire planting was infected. So that got another whole set of issues going on with another entity, and we followed up with that and subsequently with sampling of other restoration projects that were done, some of them that were more than 10 years old, and sampling, we're finding Phytophthora associated with these older restoration plantings as well. That work was expanded by the Rizzo lab, Heather Mell and Tyler Bray. So they're doing a lot of that sampling, and we participate in a certain amount of that as well. And among the 38 taxa they discovered on 28 plant species, there were both novel taxa as well as another regulatory species, Phytophthora coursina, which has not been really positively identified in the US previous to that time. So there are clearly problems with introducing Phytophthoras into the environment with nursery plants in a restoration setting. And the first reason it is not a good idea is because the restoration planting isn't going to fare very well. What we typically see is we have a lot of mortality associated with the planting material if the roots are decayed. And what happens is these roots just don't really expand beyond the original root ball. They keep decaying off, and the plants have a tendency to decline or perform very poorly. So that's one reason we certainly don't want to be using Phytophthora infested plants. The other issue is that we can have spread from these nursery plants into the wider environment, including into this native vegetation. So in this particular site here, this is the nearest planting to this native vegetation here, and within a few years after this planting was established, we detected at least one of these 5 Phytophthora species associated with the nursery stock in native species that on the site, this Melasma marina here, and that's about probably 10 meters or so away from that the nearest plant. So the movement downslope, again, the previous speaker certainly addressed this at some level. And the sort of effect over a longer period of time is we've just talked about before. This is a site in Santa Clara County, and Janelle is going to be talking about this location, I think, a little bit later in her talk. But we see a lot of Phytophthora detections in this area, and that's due to the fact that these nursery-grown plants were planted there, and after 30 years, we have an infested area that covers about 7.6 hectares, almost 20 acres, just from those plantings and spread over time, downslope and whatnot. And again, you can see the white dots are non-detections, and you can see that the infestation is really related to that those plantings. There happens to be another little couple of hits here along the shoreline associated with Phytophthora that rafted in along during a flood event there. We won't get into that right this moment. But whether it's done from restoration projects, or in this case of this botanical reserve in the East Bay Hills, landscaping from adjacent residential properties, you can get movement of Phytophthora into these natural systems where essentially can cause quite a bit of havoc. This is Arctostaphylos pallida and some other species, pallida being an endangered rare species and is being killed by Phytophthora cinnamomi in this area. From the source being this plantings in these adjacent areas here. And for those of you who are familiar with the Ione manzanita and other endangered species, Phytophthora cinnamomi is quite devastating to that ecosystem. It essentially totally destroys that plant and another sympatric Arctostaphylos species that occurs there. And we see similar things in other vegetation. Cinnamomi is one of the worst, but we have other species of Phytophthora that are out there destroying native vegetation in native forests. So clearly, there are some real downsides to moving Phytophthora into these wildland systems. Now, all the stuff we've been talking about has a kind of common thread, and that is that everything we really know about Phytophthora in these systems in California is a function of sampling and baiting these areas. And really our knowledge is limited by how much we can actually get out there and sample and bait these areas. Metabarcoding and other methods are used to try to increase throughput, increase sensitivity. But it is true that baiting is kind of a standard, the gold standard, in that you can recover the actual live cultures and you can do genetics on them, you can do host range testing on them. You aren't just working with snippets of DNA, and you know for a fact if you recovered it live, that's a live functioning Phytophthora in the area. You're not just working with some dead DNA that might have shown up there from some other reasons. And the reason also we have to sample is you really can't generally diagnose Phytophthoras from just symptoms alone, particularly soil-borne Phytophthoras. And this is kind of a good example. These are valley oaks sampled in one location that were infected with Phytophthora porcina. Actually, if you look at them, they don't really look that terrible. But one thing you do know, if you know anything about valley oaks and their growth rates and their riparian situation, a 14-year-old valley oak that's still that size has got some problems. There's no reason for these to be that size after 14 years of planting in this environment. So that's a function of the root rot that's going on. So kind of looking pre-2014, this is just our sampling. Other people have been doing sampling, but a lot of the early sampling on some of these environmental areas, native habitats, we did that. And these were areas where we recovered Phytophthora and found it affecting these native habitats. Starting in 2014, we started doing quite a bit more testing, and we and others, of planted material. And these are Phytophthora detections associated with these planted materials, mostly from, almost exclusively from restoration plantings. And if we now overlay, there's some in Southern California as well, showing here this inset. Oops. If we go forward and look at the other environmental samples taken in that period, we'll see that there's a lot of correlation between some areas where we picked up both nursery samples as positive and environmental samples in the wider area around these that were also positive. So we have plenty of documentation that's going on, and it's obviously not a good situation. So what do we do with this information? So we gather this information, and hopefully we can use that to inform the management of these different areas. In this situation of the Coyote cyanothus, that was one of the examples I mentioned there. The Phytophthora was detected at an early enough stage that it could be contained, and we actually solarized, heat treated these areas. To each planting site was removed, solarized with a fairly rigorous protocol, and that was actually quite successful at eradicating any detectable Phytophthora in those areas. So that's a potential at an early stage that you can deal with things. At kind of the far end of the stage, a last resort often is just trying to hold the ground on, say, rare plant populations. This is a situation for the Ravens manzanita in San Francisco, where Phytophthora was introduced with planting material into the very, very tiny native range. And so use of phosphite was based off of the sampling, was put it forth to try to hold the ground a little bit until some better ideas could be come up with there. And phosphite in these environmental situations can be effective if it's applied continuously. Here's a study we did in Ione manzanita. And if you look at the treated and non-treated control, you can kind of see that disease has just run rampant through the non-treated area. It's been held back in phosphite treated, but unless you continue phosphite treatment interminably, you can't keep it out. We can also, in the situation where there's nothing been done to eradicate Phytophthora, use sampling to help minimize further spread of that. And part of that is understanding where these pathogens are located, so you can direct your activities appropriately as to try to minimize risk. That risk might come from transport, from trail users, from equipment, soil movement, things of that nature. And so these can be mitigated in various ways, and there'll be some talks later on, kind of the difficulties of dealing with that. I mean, one absolute possibility at the end is also just to close areas to access. So again, Phytophthora in California, kind of the wider understanding of Phytophthora in California kind of only dates to around 2014. If we're talking about soil Phytophthora, it goes back to 2000 for Phytophthora ramorum. But other places, especially Western Australia, have been dealing with these things for a longer time. So they have another model for understanding and messaging about these things that we want to see more incorporated in California situations. So this brings me back to the guide itself. This is the title, and the purpose overall was to try to assist restoration practitioners and others in recognizing Phytophthora root rot at habitat restoration sites. And we're focusing on habitat restoration sites for a couple of reasons. One is prior to 2014, and actually until prior to the use of Phytophthora-free material, which is still not happening everywhere, this is one of the big sources of contamination in these wildland areas. Secondly, we have some identifiable targets for sampling. So this is actually a simpler system for sampling than a lot of other ones are, because at least you can identify, yes, here's some planting material, here's the root zone of that material. We can sample that. Some of the contents here we're talking about, we really start by explaining more about how Phytophthora causes disease, spreads, because really, unless you understand the dynamics of Phytophthora epidemiology in the field, it's really difficult to do effective sampling, because you're not incorporating that information you need to optimize your sampling. We do talk about symptoms such as well, we're talking about symptoms, you can see, and symptoms are kind of variable, and often there's a lot of look-alike symptoms. So these are things that have to be kind of filtered through when people are looking at that. We do talk about the whole process of collecting samples, what you sample, where to sample, when's the best times to sample, how to do that sample, and importantly, again, why are you doing certain things? That gets back to this first point. So people kind of understand the mechanisms, what we're doing here. And then we talk about baiting using pairs, and we can get into pairs later on. But the point is, for California and for a lot of species we're working out here, this turns out to be a fairly effective bait and readily usable. So we're looking to use this sampling by non-plant pathologists, partly because there's just not enough plant pathologists in the state to deal with the hundreds of thousands, if not, well, actually the millions of individual planting sites, and hundreds to thousands of areas where people might want to really have information about what's going on there. And there's a background of these types of projects in New Zealand. They actually have the Maori, the Manahuena are involved in sampling for Phytophthora agathodicida, which is the causal agent of cowrie dieback. And so there's a model for training and having citizen scientists, as it were, out there doing this kind of work. Again, one of the things is to try to key in on what to sample, and we do have, this is a table from the publication, and we do talk about symptomology a little bit there, acute symptoms, chronic symptoms. One of the issues is that most of these symptoms are really relatively non-specific. There are a lot of other things that can cause similar symptoms. So for example, if we look at this total dieback, kind of an acute symptom when the whole top browns out. In this particular sample, this is associated with Phytophthora cactuarum. These plants just have no roots. And you can also see the association of that with where the irrigation was used, but whether the irrigation's there or not, there's still Phytophthora from the nursery plants. In this other site, we have similar symptoms showing up, pretty much the same year, not a little bit later in the season. But in this case, this girdling damage was not caused by Phytophthora. We did have voles in some of these, and that they caused some of the girdling, but in fact, it was the measures meant to protect against voles, this aluminum mesh that was put around these things that ended up being incorporated into the stems and kind of girdling it, because the mesh doesn't callus over very well. And so that puts stress on the whole vascular system, and then we had Dothrella iberica coming out of these stressed plants, girdling them the rest of the way. So there's plenty more in the publication, which isn't quite out yet, but that'll be linked on the sites. The point is that I want to finish with a couple of thoughts that really we want to prevent Phytophthora introductions rather than deal with the consequences. So the idea of planting Phytophthora infested material is something we really don't want to do. There are ways to avoid that. Here's some oaks were planted where in an area where direct seeding of oaks would be superior to using the nursery stock, which happens to be infested in this particular case right in oak woodland habitat. So that's not a good situation. So the use of just Phytophthora free stock from nurseries using Phytophthora exclusion BMPs, those are put forth by the Phytophthora working group, et cetera, and the AIR program uses. The first compliant stock using these BMPs was produced in 2017, which is only 3 years actually after 2014. So we actually made excellent progress in just a few years to getting people to produce clean plant material. And testing is part of that clean plant material method. We use this leachate test method, which is a little bit different than what I think they're using in the UK. And we've explained it all on our website. We have the detailed protocols there. And just recently, we finally got this paper published. It's out at first look right now, but it describes and excruciating detail how this method is run and sort of the science that backs that up. So with that, I pretty much consume my time, and so I'm open for questions if there's time. Otherwise, I can answer those in the chat.
Oh boy, that's exciting, Ted. And so this is, we do have to move on. We don't have time for questions, but that's going to be a link provided to everyone participating here today. Yes?
Certainly.
Certainly. Okay, awesome. Well, thank you very much for all that work and presentation. And I'm very happy to introduce Dr. Del Castillo, who is going to present on an exciting new program at UC Davis. Take it away, Joanna.
Thank you, Leanne. Let me see. You cannot see my screen, right?
Not yet. I do believe we might have some time towards the end. So there's some questions I see for Ted that we can circle back to later. I believe Janice said we should have some time if people can hang up.
So my computer died last week and it was formatted last week and it's giving me trouble, but I've been able to share screens in other meetings, so. Okay.
Would it be helpful, Joanna, for me to give a question to Ted while you troubleshoot
Sure,
that?
yeah, go ahead.
Okay. Okay, Ted, one of the questions here is, what shall I do if I see weak plants in my restoration site?
Besides get all sad. The main, I mean, the idea of this restoration guide is to provide some tools and some protocols to try to sort things out. Okay, so there are lots of reasons plants can look weak. There's lots of reasons plants can show dieback and other things. Sometimes that can be due to Phytophthoras, sometimes it's not. So really the diagnostic process is trying to narrow down, try to rule out the most obvious things, and then where obvious things are kind of ruled out and it still provides the possibility to consider doing sampling and get a confirmation through that method. So that would be kind of a concept of how you can proceed there.
And send pictures to your pathologist friends.
Definitely.
Joanna, you just let me know when you're ready. If you're ready, there's enough participants and my Zoom skills are such, I'm like, not sure. There you are. Okay. A little more time for questions. Okay, there is another one here about, oh boy, it feels like a big question. So just keep in mind, Ted, that when Joanna's ready, we'll pop over. Oh, Susan is suggesting that maybe Joanna can share her slides to Bonnie and Bonnie can share them.
Let's see, Diana, you have Joanna unmuted. She seems to have some issues with that right now.
Hey, that's not my power, Bonnie.
Oh, Bonnie's got that.
Yeah.
They're figuring it out. It's okay.
Okay. Oh, wait, I see she's unmuted.
Okay, yes.
Welcome back.
Thank you. My apologies. Yeah, I learned the reason why I couldn't share screen was because it was being recorded, and the university has been very strict with security, so that's why it had a lot of settings that didn't allow me to share my screen.
All right. Well, we are seeing your screen now.
Can
Yay.
you see the PowerPoint?
Yes, it's still in the it's not, there we go. Yeah, it looks like you're getting
Okay.
into the presenting mode.
Yes.
Here it comes. There we go. Okay.
Okay, perfect. And my apologies for the technical difficulties, and thank you for your patience. Well, I'm very excited to be presenting today my new program. I'm Joanna del Castillo. I'm the new Assistant Professor of Cooperative Extension here at UC Davis. I'm in the Department of Plant Pathology, and my specialty are greenhouse and nursery crops. I have known some of you in the past, but now I'll be introducing what I'm doing as the greenhouse and nursery pathologist here at UC Davis. So the Castillo Lab is a research and extension lab. Mainly the research that we do is applied research that comes from the challenges that the nursery industry is facing here in California. And based on that, we focus on develop basic questions to understand how can we solve diseases and understanding the biology of the pathogen and how can we manage that. And also how can we produce outreach and extension materials to work closely with producers to solve these disease challenges. None of the work that I've done and I will be doing in our lab would be possible without my team. I have those two pictures here because we have different settings of lab members in my laboratory. I'm happy with we have now 4 full-time members, and we have a good number of undergrads that help us to do all the different work we do at the lab. And then in terms of project, as I'm supposed to cover all crops grown under greenhouse and nurseries, I will go into that, but I will focus now in ornamentals, which is the priority for this audience. And so one of the projects we're mainly focusing is in how to improve pathogen detection in California restoration nurseries. Later today, the postdoc in our lab, Rushika Gashab, will be presenting some updates in the research we're doing with exploring new techniques for detection and comparing the currently available ones. And also an important component that we have is the accreditation to improve restoration program, which is how can we work together with nurseries to improve their best management practices that can guarantee the production of healthy plant stock. For that, we're participating with people that are part of this meeting. Diane is part of that group, Susan Frankel from the USDA Forest Service, and Tetsuiki that just presented have been working and were the ones who initiated this program. And now I'm happy to join the program a couple years ago, and now we're leading it here at UC Davis. Another lines of research or services that we have with the ornamentals is providing diagnostics of ornamentals in the landscapes, in the nurseries, and also with commercial growers. For that, I collaborate with farm advisors. You have seen, some of you may be here. So these farm advisors from different counties send the samples to my laboratory in order to conduct diagnostics. If you are a grower, you can contact the farm advisor from your county and send samples to me. If you are not sure if there's a farm advisor that can be the main person, you can contact me directly and I can take diagnostic samples. If you are interested in sending some samples, and I need to clarify, these samples are free of charge. You can go to my website, as you can see the link there, and you can go into the extension portion of the website where you have the service in English and Spanish. If Spanish is your first language, we provide that service too. So for that, you will fill a form and send the samples to me. In order to fill that form, you have to include all the details that were considered in the nursery or in the landscape where you see the symptoms of the plant, so that give us better ideas and better clues of what may be the cause of this disease. If you're interested in submitting samples, Ideally, we would prefer fresh plant materials. We cannot take entirely dead plants because probably the pathogen that caused the plant dead in that plant probably moved to another healthier plant. And so when possible, of course, it's not always possible, but send the entire plant. If that is not possible, contact me and we'll figure it out on a strategy to sampling useful plants, parts, or soil. You can wrap the sample to protect them. Do not add any moisture in the sample because that will worsen whatever it has. And make sure to mail the samples in a sturdy container like boxes, not bags, so they can come in one piece to our laboratory. Usually we take between 3 weeks and 4 weeks for a final diagnostics. The reason why it takes longer is because we first need to let the grow a pathogen, even though this is a fungi, a bacteria, or an oomycete from the plant, and it may take some time to grow. Then we need to isolate it and then do some identification with that. Some cases we are lucky in order to have an easier sample, or we can get a sooner and faster result. And with that, I would like to acknowledge, well, all the members of my lab, all the funding sources that have made possible to work in this ornamental section. And some members of my lab will be presenting later on today some of more projects that we have. Thank you.
All right, thank you very much, Joanna. It was a flurried pace of a lot of cool information. So that's a wonderful resource for nursery growers. And so I'm going to jump right into introducing a research associate working in this new program, Dean Watson, who will be presenting on the Accreditation to Improve Restoration program. Hi, Dean.
All right, hello, everyone. Let's see if I can get my screen going here. Hopefully we don't have the same issue. Just one moment. All right, so here we are. Hello, everyone again. My name is Dean Watson. I am a research associate in Joanna's lab working on the AIR program, which is our Accreditation to Improve Restoration, running through the Del Castillo lab at UC Davis. So first, what is the AIR program? Well, the AIR program is an a voluntary accreditation program with the goal of producing Phytophthora-free nursery stock in California native restoration nurseries. Producing Phytophthora-free nursery stock is crucial to preventing the introduction into habitat restoration sites, as Ted's presentation so clearly showed. And speaking to the success of our program, Phytophthora has not been detected in any nurseries that have successfully implemented the AIR program's best management practices. So as of now, we have 21 nurseries currently participating in our program in both Northern and Southern California, 9 of which are fully accredited and 12 are in progress. I'd also like to note that 3 are just in the beginning stages, so they're not listed. And we have several other nurseries that have expressed interest, but they're not actively participating at this time. So our program is based on a couple of simple rules that we kind of summarize as start clean, stay clean. So what does this mean? Well, if you have clean inputs, so your potting media is clean and Phytophthora free, your pots have been heat treated, your plant propagules are all Phytophthora free, and your practices are clean, then your final product will be Phytophthora free. Then we have clean plus clean with a minor departure from BMPs. We'll consider it clean, but action is needed to maintain the accreditation. Next, clean plus contaminated is contaminated. So you could have clean potting media, but if you use contaminated plant propagules, your end result is going to be contaminated. And of course, contaminated plus contaminated is contaminated. And no treatment will prevent it from being contaminated. So I also like to note that we consider the ground to always be contaminated. Phytophthora spreads with water splash. And if you're ever unsure whether or not something is contaminated, just assume that it's contaminated. So how do you join the AIR program? First, I recommend that you visit our website at restoration.ucdavis.edu to review our information and fill out our contact form. Now, this website is still under construction. We're working on it, adding materials as we go, but our contact form is up and running. Once you filled out our contact form and gotten in touch with us, a AIR program representative will send your point of contact a nursery evaluation form. Fill out this form to the best of your ability, and at that point, an AIR evaluator will review your evaluation form, rating your self-reported practices for BMP compliance and flagging any areas that we think need some clarification. At that point, an AIR evaluator will visit your nursery to collect data on any flagged items, take photo documentation, and discuss any observations. At this point, we can provide any recommendations on improving your practices that we see that are clear, and kind of help move the process along a little faster. When we visit your nursery, the AIR team will conduct and demonstrate leachate testing for the detection of Phytophthora. If there's not enough time, because the evaluation can take a little while, we will schedule another visit so that we can do that testing for you. One thing that we do like to note, though, is that we expect our nurseries to conduct their own testing on a regular basis once they're accredited, but we will offer the service once a year. So after the nursery visit, evaluators will return an evaluation summary, and this summary is going to have your nursery's rating and any steps to address non-compliance. If at this time we determine that your nursery is following our full set of BMPs and there's nothing that you need to really change in order to get the accreditation, we'll go ahead and give you that accreditation then. But otherwise, if we observe any moderate or high risk conditions, we will come back with detailed steps required for BMP compliance, and we'll reevaluate your nursery once those have been addressed. So another thing just to mention is that once you receive your accreditation, it will last 2 years. And after that 2-year time, we will have to come and visit your nursery again and go over your nursery evaluation form to see any updates. So just some closing remarks. We understand that restructuring your nursery for full BMP compliance can be a barrier because it is a lot of work and there is some startup cost involved. So if you are not able to certify your entire nursery at once, we do give the option of accrediting a clean production area. So you can start small and expand outwards as you can. Next, I just like to underscore that AIR is not a regulatory entity and participation in this program is optional. If your nursery is not BMP compliant, there is no penalty for that. We're just going to come back and help you reach that compliance. And we think it's really important that we're helping to work together through this accreditation progress. We know that even incremental changes can be beneficial. Of course, we want full BMP compliance, but we're going to help you get there. We also provide yearly leachate testing for participating nurseries. And as I said, we do expect our nurseries to expect regular testing on their own as part of our accreditation. And lastly, I would just like to highlight that symptomatic plants can be submitted to the Del Castillo Lab for Diagnostic Services, as Joanna mentioned just a moment ago. So if you would like to find out more about our best management practices, we have them listed online. You can go to TED's website at phytosphere.com for the full set of BMPs. There you can also find out more about our phytosanitary tiers for our propagule collections and how to conduct the leachate testing on your own. If you have any questions, please feel free to reach out to us. Either Joanna or myself will be happy to help you. Our emails are listed here, and you can, of course, reach out to us on our website. And of course, we'd like to acknowledge all of our help on our team from Ted, Susan, and Diana. So thank you so much.
Great. Thank you so much, Dean. It's a fabulous resource to have the knowledge of all these plant pathologists to help support us in our efforts. And we're running just a little behind, so I'm just going to jump right into introducing Dr. Kashyap, who's going to be sharing a diagnostic tool with us.
Thank you, Deanna. I'll just be sharing my screen, and here we go. Hello, everyone. I am Ruchika Kashyap, and I'm a postdoc in Joanna's lab. And today I'm going to talk about another focus point that we are working in our lab is kind of improving diagnostics to improve Phytophthora detection. So we are all talking about Phytophthora, and we are trying our best to control Phytophthora in different ways and doing researches for this effort as well. So one of the things that could be done is adopting BMPs to prevent Phytophthora in nurseries, and that's what Dean talked about. And we have our AIR program, and then we heard a lot about it. And the other method that could be used is timely diagnosis, and I'm focusing on kind of improving diagnosis to detect Phytophthora. So for this objective of my study, what I am doing is I'm comparing different detection methods and substrates, both traditional, newly introduced, and other modern detection methods to see what method would be effective in managing Phytophthora under what conditions and substrates. So the most commonly used method is the leachate baiting, which we have heard about a lot in our presentation and talks today. So which is basically like you pour a specific amount of water onto a set of pots and collect the leachate in a bucket and the flow through in a bucket, and then bait it with a pear. After 3 to 4 days of incubation, your pears will develop these lesions, and these lesions are basically plated onto PARP V8 media or Phytophthora specific media. And then this culture is then sent for sequencing in order to know what Phytophthora species is affecting the plant. And the other detection method used are the immunostrips, which are readily market available immunostrips from the company Idea, which is just like a COVID test, will kind of tell you a positive and a negative Phytophthora. So when you crush your infected plant sample in a mesh bag that has a buffer and you insert your strip into it. The other detection methods used by the labs are culture-based isolations, which is kind of plating the infected plant sample and then have your culture DNA extracted and send it for sequencing to know what Phytophthora you are recovering from that symptomatic plant. And depending on the resources of different labs can also use a qPCR assay, which is a quantitative polymerase amplification assay. But each of these detection methods has their own constraints. As an example, the peer baiting or the leachate baiting and the culture-based isolations can take up to 20 to 25 days to know what Phytophthora species is actually present. But in cases, we would not have this much time, and there might be some cases we need to know what Phytophthora it is pretty sooner. The methods like immunostrip are effective, but they are sometimes non-specific and they can cross-react with other Pythium species and Phytopythium species. Meaning, if you got a positive immunostrip test, that is not 100% certain that it is a Phytophthora. It could be other species from any of these that the company claims itself that it can cross-react with these species. And methods like qPCR could be really costly. And then it again depends on the resource constraints if a lab has a qPCR or not. So we wanted to incorporate something that is fast, reliable, and can give really robust results. So we brought the RPA assay into this picture, which is the recombinase polymerase amplification assay. So our idea was to take an infected sample, crush it in a buffer, use this crude plant extract to run the experiment by using Phytophthora specific markers and visualize the results in like 20 to 30 minutes. So this is an isothermal assay. That means it runs at a constant temperature and can give you results basically in 30 minutes. So we started testing our Phytophthora species, that is 20 different Phytophthora species, 45 different isolates of these species on genus-specific markers that are already published by Miles et al. So after testing, we realized that it was really effective in giving amplification within 5 to 15 minutes. So here is some of the pure DNA that we tested from these different species, just to make sure whether all the Phytophthora will amplify when they are amplifying, and kind of setting it up. So one thing I want to point out here, that you will see different species here. This markers does not tell you what species is detected. It will only tell you whether there was a Phytophthora or not, because these are genus specific markers and not species specific markers. But in order to know whether it works on a range of species, we tried different species and saw the efficacy and the sensitivity of these markers. So what does it look like when you run the reaction on the machine? This is what it looks like on the machine. You can see if it's a positive Phytophthora, it will give you a plus sign. And if it's negative, it will give you a negative sign. And this is how you see that curve on the machine. But then when we extract the data, we run it in Excel, and you can change it to look better. So this is what the thing looks like on the machine, where you see the amplification. Here you can see the amplification is seen in 5 to 15 minutes. So here is another example that the amplification can vary between 5 to 15 minutes, but a positive amplification is seen. Negative is water, so nothing happened in our water control. We also tested some Pythium species, pure DNA, and we saw that none of our Pythium species tested amplified, and the positive control used here was same Phytophthora pure DNA, which amplified, and the negative is the water, which did not. Meaning that these markers are not only sensitive, but also specific in detecting Phytophthora. Now, we have different detection methods available, different conditions, substrates, and economic constraints. So the question is what to use and what could be actually effective in using. So this study was conducted by Joanna on coffeeberry plants infected with Cactorum. So she infected the coffeeberry plants in a greenhouse with Cactorum and tried to test different traditional detection methods over a time point. And this is on the x-axis, you will see different time points of sampling and proportion of Phytophthora detected in each of the time points. You will see that leachate baiting is really effective in detecting Phytophthora. It doesn't matter what sampling time point it is. And root immunostrip seems to, the detection percentage of Phytophthora seems to increase over time. This could be related to pathogen biology that, after infecting, it takes a little while to actually cause infection and cause symptoms. And immunostrips were really inconsistent in detecting Phytophthora. So based on this study, the question we had was, is Phytophthora detection affected by host, timing, pathogen species, detection method, substrate? What is the Phytophthora detection looks like in all these questions that we have. And Joanna just tested it on one host and one species. What if we included more species and more hosts and compare the detection methods? So for this experiment, we planted a randomized complete block design and had 2 crops, coffeeberry and Teon, and then infected the plants with 3 Phytophthora species, Cinnamomum, Cactorum, and Cryptogaea. And we also included a non-treated control and sampled the plants at 3 sampling time points, that is 6 weeks after inoculation, 12 weeks, and 18 weeks after inoculation. So this is how the plants were arranged in sets, and then different detection methods were performed. So this is what the picture looked like, where inoculations was done on this date, and these were our sampling time point dates. And then at each sampling time point, we detected or compared leachate method, direct route isolations, immunostrips, RPA, and qPCR at all these 3 sampling time points to see what the picture looks like. We have already finished our detections from immunostrips, culture-based isolations, and pairs, that is leachate baiting. And I'm going to show you some of the results here. And the samples are stored in -80 until we perform any other tests like qPCR and RPA. So you will see here the same concept where along x-axis you will see the sampling time points and the different methods of detection, and the bars will represent the Phytopthora species that was detected. Here you will see that leachate baiting, again, similar to what Joanna saw, that was significantly effective in detecting Phytophthora across different Phytophthora species and different time points as well than the direct root isolation and root immunostrips. Where root immunostrips and direct root isolations were very inconsistent in detecting different Phytophthora, even though it seems like there is an increased trend in detection over later time points. So comparing the root immunostrip and pair immunostrip, basically what we did was instead of putting the root inside the immunostrip bag, we put in the pair lesion that is developed after leachate baiting inside the immunostrip, and we saw that changing the substrate affects the detection as well. Same immunostrips, we can see pair immunostrips are really good in detecting Phytophthora comparing to the root immunostrips. And we saw similar results in Toyon as well, where leachate baiting and pair immunostrips were really effective in detecting Phytophthora than other two methods, but we saw a higher detection in the other two methods than compared to what we saw in coffee berries. So what can we conclude from this conventional assay detections? We can conclude that among the conventional detection methods, really leachate baiting and pair immunostrips had significantly greater detection than doesn't matter what host it is, doesn't matter what timing are we trying to detect the Phytophthora. And detection of all Phytophthora species were really effective in controlling the false positives. So it is really, all the methods are really good in eliminating the false positive. We're currently working on testing RPA assays on all these three sampling time points and qPCR assays, and I'm going to show you some preliminary results of the RPA assay as well. So we have started working on testing the RPA assay on our samples that are stored in the -80. And then how does this assay works? Basically, we take like 0.5 gram of the symptomatic roots and then take general extraction buffer from this company, Agdia, and then put the buffer and the sample in the mesh bag and crush the sample and take that crude plant extract in testing. So here you will see, this is what the preliminary picture looks like, where we are testing plant samples infected with Cinnamomum, Cryptogaea, non-treated control. Our positive is the pure DNA and our negative is water. So these are the sample from coffee berry at time point 2 sampling. So we saw that our positive is amplifying and our negative did not amplify. So this means it is a valid run. And when we see the Cinnamomum detection, we see detection, but at a very later time point, and we did not see detection in the non-treated control, and we were not expecting it as well. Now, this later amplification could be due to, when it's in a crude plant extract, the pathogen load is really less. So when there is less pathogen load as compared to the pure DNA, you will definitely see a later amplification. But according to our threshold that is we have set up, this is a positive sample. But we did not see any amplification for the Cryptogaea inoculated samples. So we are trying to play around with the primer probe concentration to improve the sensitivity of detection from the crude plant extracts as well. And hopefully we will have better pictures next time. So we are hoping to develop, validate a successful RPA assay and determine the most effective method in detecting Phytophthora depending on sampling type, method used, and species, and recommend an effective approach for both nursery growers and, the labs, depending on what resources they have. I want to thank all the funding agencies that supported this project and all my lab mates from the Del Castillo lab who has helped me in sampling and all other processes. And I would want to acknowledge other collaborators who have helped me troubleshoot through this process as well. And any questions? I don't know if we have time, so but I'll be.
Thank you so much. I imagine there are questions. I have questions, but we don't have time. What it's time for me now is I'm very happy and honored to turn this over to Mia Ingolia, who is going to moderate the next section of talks and discussion. Take it away, Mia.
Thank you, Diana. Okay, so next up we have Janelle Hillman. Janelle is a senior biologist / vegetation ecologist from the Santa Clara Valley Water District, and she has been managing Valley Water's plant pathogen response program since 2015. And today she is going to talk to us about Phytophthora and large construction projects. Go ahead, Janelle.
Thank you, Mia. Good morning, everybody. Are you able to see my screen?
Yes.
Okay, so I just want to say how incredibly inspiring it is to hear so much good work being done through the AIR program and at all of our restoration nurseries. I'm pretty thrilled that in the project that I manage at Valley Water, we do conduct leachate baiting of all of our contract grown nursery material before delivery for our mitigation projects. And in 6 years of testing plants and contracting and partnering with our BMP compliant nurseries, we've conducted over 500 tests of almost 28,000 plants and not a single Phytophthora detection. So it's really exciting on this end that we're restoring our mitigation sites in a good way this way. So what I want to talk about today is what do we do on some of these more legacy sites where we already have introduced Phytophthora contamination. We might have projects in these sites or other work activities. So how do we manage these areas for soil-based Phytophthora in non-forested systems? So the project I'm going to talk about today is the Anderson Dam Seismic Retrofit Project, located in South Santa Clara County in Morgan Hill. It's the largest reservoir that Valley Water manages. And in fact, 10 of our other reservoirs would fit inside of Anderson Reservoir. It has a storage capacity of 90,000 acre feet. So it's really huge. It was built in 1950, and in 2011, it was determined to be seismically unstable. The Calaveras Fault lies within 2 kilometers of the dam. And our early seismic stability studies indicated that in the event of a 7.25 magnitude earthquake, the entire dam would liquefy and a wall of water 35 feet high would hit Morgan Hill and Gilroy down in South County, and within a matter of hours, travel north to San Jose. So the current project is expected to be completed in 2032. And this project is really the perfect storm of a lot of different issues that we have here. So it's a fast track project for public safety, but we also have the co-occurrence of federally and state listed endangered species, serpentine soils and habitat. Serpentine is actually the state rock of California. A lot of rare and endangered plants occur on serpentine. And in fact, this population of Coyote ceanothus was bisected by the construction of the original dam. We also have 13 documented species of Phytophthora located in the project area. And I think as Ted sort of touched upon briefly earlier, these were the result of a revegetation project back in 1993 when we enlarged our spillway. So this is the current project area. This is what the site looks like right now. And before we're actually beginning the seismic retrofit of the dam itself, after the Oroville issue that we had a few years ago in Northern California, we have a new order. We actually have to create an enlarged outlet through the dam before we can even start to work on the retrofit of the dam itself. So that's actually a separate project, but we're creating this larger outlet. We have hundreds of workers on site daily from at least 6 different companies. This phase of the project is 2 to 3 years of work. And this picture here, this is the abutment of the dam. And this is where we have the most highly contaminated portion of the project area. This map just shows the current work area overlain with some background work that was done by Phytosphere Research to look at potential contaminated areas in this project area. So the red circles are positive soil sample collections of different Phytophthora species. The white dots are negative sampling results. And then we do also have some triangles. So these are water collected samples here. This is a view of the larger project area showing positive and negative soil samples as well as water samples. The dam abutment itself has 6 different species of Phytophthora, as well as numerous detections throughout the rest of the project area. This is really complicated work, and kudos to Phytosphere for helping out and doing all of this field-based sampling. But I also want to put a shout out that we really need to focus on growing our new generation of field pathologists, because this is work that we definitely could not manage this project effectively without pathologists who can go into the field, understand how to sample, where to sample, and then how to interpret these results in a way that we can use to guide our construction activities. It's also complex permitting for this project. So Anderson Dam is actually a federal hydroelectric facility. So it's regulated by FERC, or the Federal Energy Regulatory Commission, as well as DSOD, Division of Safety of Dams. And we have 2 FERC orders related to Phytophthora that has been found in the project area. We had to prepare a Phytophthora pathogen management plan, which includes BMPs during construction. And we also had to prepare a monitoring plan, which involves both pre-project baseline sampling as well as post-project monitoring. And this is to ensure that we are not spreading Phytophthora contamination to new areas outside of the immediate project, both downstream onto county parks property, and then upslope in the reservoir, which is where the majority of the population of the federally endangered Coyote ceanothus occurs. So some of our project challenges, and there are many, this large stockpile in this picture was actually excavated from a portion of the dam abutment, and we're actually tunneling through the dam itself right now to create this larger outlet. So this stockpile of soil or dirt, it contains both contaminated sediments and non-contaminated sediments. And it was very difficult in this project to determine how to keep these contaminated soils separate from non-contaminated layers. So we don't have a lot of space to stockpile soil. It's extremely expensive to off-haul soil and dispose of it if it's contaminated. And a lot of this soil, actually all of it, is needed for the ensuing Anderson seismic work itself. We're going to need to place all of this soil in reservoir to build a large cofferdam and other features as part of the project. So we really need all of this soil, unfortunately. We're also on a fast track timeline to completion for public safety. And the original pathogen management plan was, because this all happened so fast, with the addition of this tunnel boring portion after the Oroville issue, the Phytophthora plan was added as an addendum to our contract specs, but there was no specific contract spec language drafted. So I've actually had to spend quite a bit of time translating that original Phytophthora pathogen management plan into contract specs that the contractor can actually understand and implement. So we have designated the site into areas that are clean and dirty. We have signs up. We're trying to guide workflow efforts. We conduct environmental awareness trainings before each new phase of the project starts. As I mentioned, there's hundreds of workers on site from various different construction companies, from drilling to engineers to construction. It's just a madhouse. And the worker environmental awareness training is a real key part of managing this issue. The workers on site actually call Phytophthora phallopia. That's like their joke term for what this issue is. But frankly, I don't really care what they call it as long as they understand what our BMPs are and what we need to do to prevent it spreading. We have boot wash stations, 70% isopropyl alcohol, etc. There's some special nuances with the project, such as a fish relocation effort that we've had to do over the past 2 summers. This is occurring in the North Channel, which is just downstream of the dam and dam abutment. And half of that channel has been designated as dirty and half of it is clean. So we have some really specialized BMPs for relocating fish when we dry back the channel and then placing them farther downstream. You see this clean tarp here. This vehicle is rolled up to the very edge of the clean area, and all the staff to the left of the photo are working in the dirty area. So there's a whole workflow associated with that. We conduct equipment inspections before any new equipment is brought on site. We don't have a large formal wash station, but we do use smaller power wash stations. That's what this green sort of structure here is in the photo. And what this is showing here is the path crossing from the contaminated side of the North Channel to the clean side of the North Channel. So what we did was lay down this crane mat, which is this wooden platform here, and then equipment rolls onto the platform from the dirty side, is power washed here and cleaned, and before it rolls off of that sort of transition area into the clean upland beyond that. So this is really what we're trying to protect. This is the federally endangered Coyote ceanothus. The largest occurrence is at Anderson Dam and Reservoir. And here on the right is a group of tule elk on the shoreline of Anderson Reservoir. And just some of the issues with this project, we don't really know. If you have this large stockpile of dirt, how do you test that stockpile? Because what I constantly get asked is, well, which part is clean, which part is dirty? How do we remediate it? In areas that are contaminated, how deep is that contamination zone? Can we scalp that layer of soil off and dispose of it? But how deep do you go? If we're using this large stockpile of soil in reservoir, how do we cap it to keep it from mobilizing at the end of the project to adjacent areas on the shoreline? So these are just a few of the really large questions that we're trying to tackle. And honestly, I don't think there's a lot of answers, but it just helps us to work on projects like this to kind of further refine our understanding of how Phytophthora moves in the environment once it's introduced into wildland or urban interface areas. I don't know if we have time for discussion, but we can chat offline as well if not.
So we're running pretty tight on time, so I'm going to have Joanna do her presentation, and then if people want to stay, we can keep the meeting open until noon and have a discussion and answer questions. So thank you so much, Janelle. Next up, we're going to have Dr. Joanna Del Castillo, the Assistant Professor of Cooperative Extension at UC Davis, talk to us again, and she's going to talk about risk-based sampling for Phytophthora in nurseries.
Yeah, thanks so much, Maya, for the introduction. So this presentation is based on a question that we recurrently have when we visit nurseries and when we talk with nursery growers. So when you are selecting plants for Phytophthora testing, how do you select? How do you decide which plants to tackle? And so, yeah, so we'll go over this first question, which plants should I test? And a question that we get also all the time is, how many plants should be tested? So to start answering the first question, which plants should I test? Definitely don't pick randomly the plants. Start looking at the different crops you're growing and look for symptomatic plants. If you see anything unusual with the plants you're growing, if you see some low vigor, if you see some wilting, stunting, dieback, plant dead, definitely those are areas where you want to focus on. These pictures are an example of Cyanotus, coyote mint, that show different symptoms of branch necrosis, wilting, dieback, and this is an example of a root rot from one of these plants. So definitely start by looking in unusual plants, unusual symptoms that you see in plants, and those are the plants that you want to prioritize sampling. Again, this is more examples of how symptomatic plants look like. This is a patch of epilobium plants that we observe at a nursery. And then besides looking at the foliage part of the plant, look at the roots, how the roots look like. In here, we had an example of a healthier looking plant. As you can see, these white colors are the roots, so it's actually healthy looking root ball compared with a pretty diseased, pretty rot roots in a symptomatic plant. So it's always good to compare how a healthy plant should look like and how a not so healthy plant looks in order to discern which plants you wanna further select. So something that is very usual with Phytophthoras is that infected plants can remain asymptomatic for long periods of time. The picture that we have here is a bay laurel that we purchased for some experiments, and before doing any experiments with those, we did some leachate testing, and we found over 3 different Phytophthora species infecting these bay laurels. So definitely Phytophthora infected plants can remain asymptomatic for long periods of time. So how do I select the plants if all my plants in the nursery look pretty healthy? So then we will propose to select plants based on the risk of contamination. So we'll start by different risks based on different scenarios, starting with the plant propagule source. So what will be a high risk if you have incoming plants from another nursery that are not a BMP compliance, definitely that will be a high source of contamination. If you take plants from propagations from cultivated landscapes, if you don't know the source of the plants from these cultivated landscapes, or if you take seeds or cuttings from areas that were known to be infected with Phytophthora. So definitely that'll be high-risk plants that you for sure would like to test. What would be a low-risk Phytophthora? Low-risk plants will be plants that are grown in nurseries following best management practices. As you can see in here, plants are on a raised bench. The propagules that you collected were high from the ground, were collected from areas that were not infested with Phytophthora, and ideally in native remote places. Continuing with high propagule type, if the propagules that you pick for propagation are in contact with the soil or at a pretty low distance from the ground, or were collected during the rainy season, which we don't recommend to that, those definitely will be high risk propagules. Versus low risk will be clean seeds that were collected ideally from above ground or tip cuttings. If the seeds have some disinfestation treatment when they were stored, those will be low risk scenarios of plants having a Phytophthora. Now, once these propagules are growing and once you have plants in the nursery, how can you select what will be a high risk? So definitely plant age, older plants that have been for more than one growing season in the nursery, they have a higher likelihood that if Phytophthora is present, they may get contaminated with those, versus younger plants that those will be a lower risk, even though let's consider like if this was the first season in which the plants were growing, and if you're keeping clean conditions for growing them, there's a lower chance for them to be infected. Also, where are these plants within the nursery? So for instance, the plants that will be high risk of having Phytophthora are plants that are close to sources of contamination. For instance, if you know you have infected plant material and you have some plants close to them, definitely that's a high risk. That's why it's important to keep enough distance between the different blocks or group of plants that we have. If you have plant material that is not growing on the nursery, again, because you don't know the conditions in which those nurseries were from. And then if you have plants that were pretty close to water courses, exterior fences, or entrances, definitely those are higher risk of contamination from external sources or from water splash. Lower risk plants, again, are plants that are well separated from any source of contamination. Now, in terms of plant species, well, it is known that Phytophthora and root rot Phytophthora has a pretty wide host range that can infect hundreds of plant species. So definitely there's many high-risk plants out there, but ones of the highest risk are flowering plants or dicots, such as shrubs, trees, or bedding plants. These are just some examples of the hundreds of species that can be susceptible for Phytophthora, but these species that I'm listing here have been some that we know are pretty regularly growing by nurseries, And based on some CDFA surveys done after Phytophthora dentaculata outbreaks, these species harbor highest number of Phytophthora species, such as the manzanitas, the cyanotus, the cistus, the sticky monkeys, the toyon, the coffee berries. But again, I want to make emphasis, not only these species, many other flowering plants can be a high risk of infection too. Conifers also can harbor Phytophthora and can get symptoms from Phytophthora, and some non-grass monocots. And then low-risk plants, grasses are low risk of having any Phytophthora infection. Has been documented one association of one Phytophthora species with grasses, but that's not something that we regularly see or has been reported in the literature, as well as ferns. Also, another consideration to take into account into which plants to select for testing is where are these plants going. So if you're growing a plant material that would go to pretty sensitive planting sites, such as very rare habitats or close to sensitive species or in pristine locations, these plants should be tested at the highest rates, because of course we don't want to bring any contamination source to pretty sensitive or endangered ecosystems. So you should definitely prioritize these plants if you know you're going into sensitive areas. So how can we mix, or how can we make decisions based on what plants to test? So basically start by picking symptomatic plants. Pick older plants that are known to be a Phytophthora host. If plants are close to sources of contamination, or if their propagation material collected from soil or plants coming from other nurseries, definitely those are the ones of the higher risk that you wanna sample for sure. And then if we understand like the logistics to test everything, that's not feasible. So if you need to choose what not to test, then select the low risk scenarios. Don't necessarily need to test all the grasses or young plants that you know they have been grown under the nursery Phytophthora based management practices. If you start from clean seeds and all clean conditions, there will be a lower chance that those plants may have Phytophthora. Now, going to the second question that we also get is like, how many plants should I test? Well, for us as pathologists, ideally, if every plant could get tested, that would be ideal, but we know that's not feasible. So definitely there's not a set number of plants to test. Phytophthora detection can be affected by the sampling method, as we saw in some of the presentation. Also, it can be affected by the infection rate. The lower infection level there's in your nursery, the more plants you will need to test in order to get a positive result. And then also, depending on the batch size, test as many plants as possible. So if you have a small batch of plants that need to go to a restoration site, ideally try to test 100% of the plants. And then if you need to set, if you have larger batches of plants for testing, select the plants based on the risk of contamination that we went over. So just to summarize, select the plants based on the risk of contamination and based on the symptoms observation. Use a detection method that had higher likelihood for Phytophthora detection. So the lead sheet baiting method has been the method most effective at getting different Phytophthora species from different hosts. And then in case you're testing an individual plant, one, you can send it to my diagnostics lab, or in the nursery, you can do a bait testing from the symptomatic planter. In this slide, you can see some of the resources that have been put out by Ted from Phytosphere into like this risk-based testing. And with that, if there's time, I'll be happy to take any questions.
Okay, thank you so much. If you guys want to start, if people want to start dropping questions into the chat for either Joanna or Janelle, I can start fielding questions. It looks like there's a question for you, Joanna. What do you think of detection with molecular tools, metabar coding and eDNA? Is it feasible? Is it robust enough?
Yeah, definitely metabar coding is a great tool in order to get to get a broad view of what species are present there. One thing, for instance, like one con with using metabarcoding for diagnostics is the markers. So the markers that have been regularly used for Phytophthora detection have been the ITS 6 and 7 markers. These markers, in some cases, don't have resolution into the species level. In many cases, you can get only up to a cluster, and you won't be able to differentiate, for instance, if you have a Phytophthora ramorum or another closely related species that are within that clade. Nevertheless, there are some efforts, and there's another marker, it's like the RPBS marker, that claim to have pretty higher resolution than the ITS 6 and 7. So that will be a metabarcoding marker that could be used in order to have like this species differentiation. But one thing with the metabarcoding too is like depending in what is your substrate that you use to take the DNA from, you are not certain if all the species that you got from there are active and are being a pathogen. So I would say metabarcoding approaches definitely help us to get a broader spectrum of what species are present, definitely can be more sensitive, but I will always follow those with a baiting or a culturing approach in order to confirm that the organism there is biologically active.
Okay, we have another question for you. Diana's asking if the AIR program will be able to provide the risk-based guide in Spanish.
Yes, I can do it. Yeah, and then like these risk-based testing, these materials will be uploaded in our AIR website, and definitely I can work in having this version in Spanish so it can be accessed by Spanish speakers at the nurseries. Yeah.
Okay, we have a question for Janelle. Janelle, have you considered using compressed air for cleaning heavy equipment? Collaborators up here in Humboldt have tested this, and it seems to be at least as effective as water for getting big clumps of soil out of tracks.
Yes, we are considering that and probably will end up using it. The formal wash stations that are typically used on really large projects and at landfills are just way too expensive. They're like hundreds of thousands of dollars. So yes, we're looking at alternative methods such as small scale power washers, air compressors, anything that we can do. It takes a little bit longer when you're using these smaller pieces of equipment than if you had a formal wash station where you're just rolling onto the equipment, it's automatically activated and then you roll off. So for the actual seismic project itself, if we have hauling of like hundreds of trucks a day, I don't know if we might move to that more formal method. But for now, yes, these are the methods that we're looking at and implementing.
I'm curious if any of the plant pathologists in the meeting can weigh in about what Janelle should do with her huge pile and figuring out how to mitigate for that and what your recommendations would be.
I've been thinking of that, and I have a question for you, Janelle. Would it be even feasible to try to steam that dirt, or is just too much of a volume to do it?
It's just so large. It's like hundreds of thousands of cubic yards of soil. It's like stories tall, and it's constantly being added to. So yeah, I don't think that would be a feasible method to eliminate. I don't think we could heat it that much. What we're trying to do is separate the more contaminated surface sediments from work in the channel and place those in a separate stockpile. And then when we're boring the tunnel through the dam itself, which is, you know, really deep subsurface soils, we assume those are not contaminated. So we are trying to separate somewhat the piles. And then when we reuse that soil in reservoir, trying to put the most contaminated sediments down first and then capping with relatively cleaner fill. But again, there's not a lot of space on the project's site to stockpile. So that's one of the major issues.
If I can chime in, yeah, we're working on every idea we can come up with, but when you're talking about the scope of how much material there is, the relative lack of control there is in terms of dealing with all these contractors and all these processes going on and things happening before you can actually do anything about it, your options are really limited. So it's kind of a triage situation to try to figure out, okay, what can we do to minimize the risk, given that we've got really a giant, as Janelle, I like the term madhouse, but yeah, it's pretty gnarly situation to work with, and there's no cut and dried sort of answers.
And with a big pile of soil, I mean, we're trying to keep it clean or diluted as much as possible, but how much dilution is enough, right? Like, at what level is there such a small amount of inoculum that maybe it's a lower risk? Because all of this is just risk evaluation and trying to just minimize risk as much as possible. But I just feel like there's so much we don't know about Phytophthora in the soil, and you know, is it going to mobilize when we put it in reservoir, and you know, a lot of just the issues sort of related to that.
I like the
Yeah.
idea of burying it deep, though. Seems like that would make it a little safer.
Yeah, and then I have a question. So when you say like you're trying to dilute the infested soil with the non-infested, have you started or thinking about testing that diluted soil?
If there was a way to do that, but it's just a big pile of dirt. So if there was a way to do that effectively, that would be really helpful.
Yeah, the problem is that we were talking about such vast quantities that, and we don't actually know that inoculum loads even in our most contaminated material, and we just know that we could detect it in those areas. It's also very heterogeneous. So, you know, you could go take a sample somewhere, and you know, a meter away from that could be a hot spot that have 50 times the amount of inoculum, or you know, you could have 0 versus, you know, tons. So it's not like we have this all mixed into a container. We can get a uniform mixture of anything there. So it's super challenging in terms of just the dynamics of even trying to deal with such huge quantities and all these uncertainties. But yeah, I mean, in terms of the long-term result, what we would like to do is put most of it in deeper layers of fill, if possible, the most highly contaminated material. So the real trick is, okay, can we still differentiate between our more contaminated and our less contaminated material based off of this kind of less than uniform methods of putting soil on this giant pile. So there are some efforts right now, as you know, maybe didn't quite get into, we have some more highly contaminated material that has to come off, and the idea is to try to maybe segregate that into its own pile, so at least we don't have to deal with that extra blob of high contamination in the big pile. But it's a hard mess to deal with. Just put it simply.
All right, do we have any other questions? I don't see anything else in the chat. Oh, wait, here we go. Does the baiting with pairs work for outdoor field settings? Wondering if any of the sampling methods presented can be used in outdoor urban forestry settings.
Well, I'll chime in on that. Certainly, and actually this guide we're talking about is essentially the use of baiting method on collected root material, primarily roots more than soil. But you basically, I guess there's the follow-up questions about whether you would place buckets under bay or tan oak. There's other ways to set trapping, spore trapping for Phytophthora ramorum if you wanted to do that. There's a couple of different methods that have been tested, and those are applicable. Pair baiting in that situation is a little bit problematic. You don't really want to leave pairs in the field because they're kind of an attractive nuisance to raccoons or things like that might decide to eat the pear. So really, you really want to take the sample and then process it under controlled conditions in your lab. That's where you're going to insert your bait. And in many cases, you have to do some pre-processing of the sample to optimize the chances for getting sporulation, which is what you need to really get a detection by baiting. But actually, that said, everything we're using here on restoration plants, et cetera, is certainly applicable to landscape settings, urban forestry settings as well. I mean, if you can sample the roots, you can do the same sorts of tests.
All right, I've seen this question before. Do we know how many of these species of Phytophthora are native to California and naturally occur in the soil?
Once more, I'm going to chime in first. The bottom line is, I think we make the assumption that almost none of these soil Phytophthoras are native to California. There's been some recent work that sort of implies there could be some possibilities for some soil-borne species that might be native to California. The only problem with this is that, we're lacking a lot of resolution in terms of when things could have been introduced, given that we have 400 years worth of settlement and movement of material around the globe. There was a global trade route that ran from Asia across the ocean to California for 300 years, the Manila galleons. So the possibility that there were even early introductions hundreds of years ago into areas can't be discounted. And so with that and diversity and not understanding the origins of all these things, it's just very difficult to say. But the bottom line is we wouldn't see these species causing problems in the areas that they do if we thought they were all native species. The species that might be native, but even still could be naturalized, are all these waterborne species. But they move so readily, there's no guarantee that even which ones of those are native. But people always want to distinguish between that. And really, when we sample out in places like Janelle's area there, we're out in the wildland areas that are not close to sources of contamination. We just don't detect Phytophthoras there. We detect it in the source areas where contamination is clearly either occurred or has a high likelihood of occurring.
Thank you, Ted. All right, we have one more. For practical purposes in nurseries, is knowing the species of Phytophthora important? Should nurseries deal with different species differently?
I know Joanna wants to first crack
Yeah.
at this one.
There you go. You were on mute.
Yeah, that's an excellent question. So, so working with nurseries, with native nurseries, and like you have your growing plants that it would go into a restoration, a habitat. So there's zero Phytophthora tolerance. So basically, it means it wouldn't matter to know what Phytophthora is present, because either if there's a yes or no answer if you detect any Phytophthora. Nevertheless, the importance of knowing what Phytophthora we have is important. The main importance will be Phytophthora ramorum, just to make sure your nursery doesn't have a quarantine pathogen. And then looking at the other group of Phytophthora species, the importance to know what species are present is to help us get a better idea into what are the species that have a greater prevalence and are more associated with certain hosts, and also to see what symptoms or what is the characterization of what we're seeing in the plant. Also, another important thing to mention is that, we know Phytophthora is associated with a wide range of hosts in native California plants. And a question that we have is how devastating or how virulent are all these different Phytophthora species? Again, yes, we should worry about all of them, but then one underlying question there is like, what are like the highest, well, we know which ones are the highest risk, and then like of the less common Phytophthora species, how pathogenic or how virulent they can be in a wide range of plants. And of course, we also need to consider that once these plants leave the nursery and they go into a restoration site, they may be under stress conditions, right? So there can be things such as drought or a flooding event, or many other abiotic conditions that can make the host more susceptible and then trigger pathogenicity of whatever Phytophthora species you have there. So so basically, for practicality, a nursery in theory shouldn't care about which Phytophthora species it has, in case basically if you have it, you cannot keep working with those plants, but also you want to know what species are present in the case it's a Phytophthora ramorum or not. And then for research purposes and for documenting what species are present, we want to know what other species of Phytophthora are present there.
And I would just add one last thing to that, is that if you actually have an idea of the species, I mean, if it's something common like cactorum, it's not going to give you a lot of insight, but say it was some clade 6 Phytophthoras, they're mainly waterborne or something, you might actually be able to get some ideas in terms of the rarity of the species or what species they were, what might have been the potential source of contamination, so that when you're trying to trace down in your clean nursery, what, how did this contamination get in here? It might help you identify that, yes, it maybe came in through this source because these are this species represented.
Okay, I'm not sure. Janice, do you, oh, one more question. During pair testing, can pair lesions then be put on into Agdia test strips?
Yeah, I can answer that, and Rishika's here, she can help answer that too. So what we found based on our research is that when we did the immunostrips from the pair lesions, the immunostrip tested positive at all times. We knew Phytophthora was present there. But so definitely immunostrips based on our research are better Phytophthora predictors than if you do the immunostrips from roots. Nevertheless, we know that the immunostrip can cross-react with a Pythium species. So let's say if in your pair you may have Pythium there, and then still you cannot discern if your immunostrip tests positive for either Phytophthora or Pythium. So I would say the best bet there would be, if you have a pair, you can send it to my laboratory and we can do a confirmation of if Phytophthora is present or not. But definitely the immunostrip from pair can be an indicator that something can be happening, but definitely not your final diagnostics result.
Before we head out for the end of the meeting, Johanna, could you please tell us about the plans for the spring field tour and meeting?
Yeah, definitely. So we're planning for the spring having a workshop here at UC Davis. Where we will be talking about, of course, more Phytophthora, but we'll have some of the hands-on experience that will let you guys look under the microscope how a Phytophthora look like. We'll have some symptomatic plans. We'll have some baiting for you guys to do, and showing any updates with our RPA assay that we're testing. And as well having some tours at the UC Davis Arboretum and also the Foundation Plant Sciences facility here on campus. And also we are planning with Sebastian Fajardo, that he's been working with Los Angeles National Forest to do another workshop in Southern California. And this one will be held at one of the nurseries, where we will have also like a hands-on experience for nurseries to participate.
Great, thank you all so much. I appreciate everyone sticking around a little bit longer. We're coming right up against noon, and I want people to have the time honored for that. So I'm going to thank all of the presenters and the moderators for helping make this a really informative and interesting meeting. I know that there's probably still questions we could continue to have discussion around. I will be sending out the link to the recording when it's able to be posted online. So you will be able to review all this and send this out to other people who could not join us today. And if you have any other kind of follow-up questions or issues that you wanted to bring forth and you didn't have time, you can email me and I will make sure that those questions get to the correct presenter so that we can kind of wrap up everything. But with that, I'm going to conclude this year's meeting of the CalPhytos group. I thank you all for your time. Have a great rest of your day.